Since we had started to discuss determinants of bacterial pathogenesis, which comprises of how transmission occurs, then adherence of the bacteria to the cell surfaces, and invasion, inflammation, and intracellular survival occur. We discussed this in the previous lecture. Today we'll start about toxin production and immunopathogenesis, and then after that we'll discuss how these bacterial infections, some of them are associated with cancer, and different strains of some bacteria, how they produce different diseases and they manifest differently because of different phenotypes, and what are the typical stages of an infectious disease. And we'll also come to Koch's postulate about if the organism isolated from the patient actually causes the disease. So, toxins. What are toxins? These are poisonous substances produced by microorganisms. And toxins are one of the primary factors in causing disease by a microorganism. There are 220 known bacterial toxins of different types, and 40% cause diseases by damaging the eukaryotic cell membrane, that is, the human cells. And when these toxins are coming in the blood, it's called toxemia. And toxigenicity is the capacity of the microorganism to produce toxins. Mainly, toxins are of two types. It can be exotoxins and they can be endotoxins. Exotoxins are the ones which are secreted outside the bacterial cell, and endotoxins are part of the outer cell wall of mainly gram-negative bacteria. So this is the main two types of toxins. Here again, the two types of toxins. As you can see, the left side, the exotoxin are being secreted outside the cell. Here. While endotoxin, which is part of the outer portion of the cell wall of mainly gram-negative bacteria, they are liberated when the bacteria die or when the cell wall breaks apart. So the major difference between endotoxin and exotoxins. Endotoxin is the integral part of the cell wall, and mainly endotoxin is lipopolysaccharide. Lipid A portion of the lipopolysaccharide is toxic. It's a heat-stable molecule and it is antigenic, but it's questionable immunogenicity. Toxoids cannot be produced by endotoxins, and many effects on the host are produced by only these gram-negative organisms. While exotoxins are released from the cell before or after lysis, and they are protein in structure as opposed to lipopolysaccharide. These are heat labile, that means on heating the organisms or these toxins, they become ineffective, and they are antigenic and immunogenic in nature. That's why they are used to produce toxoid. Toxoid can be produced and specific in host on causing and produced by gram-positive and gram-negative both organisms. So these are the major differences between exotoxin and endotoxins. Try to remember these because these are usually asked in Viva as well. So coming again to exotoxins, these are mostly seen in gram-positive bacteria, and the most gene that encodes for the exotoxins are located on the plasmids or phages. There are three types of exotoxins. They can be cytotoxins, which kill the cells, for example, diphtheria toxin produced by Corynebacterium diphtheriae. It can be neurotoxins, for example, botulinum toxin, which interferes with the normal nerve impulses. And they can be enterotoxins, which are produced in the gastrointestinal tract. An example of which is cholera, which is produced by Vibrio cholerae. So, cytotoxin, neurotoxin, and enterotoxins are the three main types of exotoxins. So, what is happening after the toxins are released? What is the response to toxin? If the exotoxins are released, the antibodies against the toxins, antitoxins are produced by the cells. And these exotoxins, when they are inactivated by means of heat or by formalin or phenol, they no longer cause disease. That's why we said these are heat labile. But they can also stimulate the production of antitoxin. That is why this property is used in forming toxoids, so that these toxoids can lead to production of antibodies and they can become less. Then the toxoids, these are the modified toxin by heat, chemical or radiation that have lost their toxicity, and they are injected in terms of vaccination to stimulate the production of antitoxins and produce immunity. Example is DPT vaccine, which is a diphtheria, pertussis, tetanus vaccine, which is given in the childhood vaccination, and it provides protection against Corynebacterium diphtheria, Bordetella pertussis, and Clostridium tetani. And these three are toxoids. And this is part of required immunization of EPI. EPI is expanded program of immunization, which is given to every child when they are born in the first 1 year of life. These immunizations comprise of diphtheria, pertussis, tetanus, measles, mumps, rubella, and polio. All these are part of EPI. But out of these, which are toxoids? Toxoids are diphtheria and tetanus. These are toxoids. The others are different molecular structure which are used for immunization. Even for vaccination, which is given to every child, childhood tuberculosis is for that there is a live attenuated tuberculin bacillus is given. For rotavirus is another viral disease. For that, vaccination is also done. And out of these vaccinations, only diphtheria and tetanus are the toxoids. Most genes that code for exotoxins are either plasmids or phages. Phages, remember, it's the virus that infects the bacteria. And by means of lysogenic convergence, they can produce exotoxins. And these cytotoxins inhibit protein synthesis, resulting in cell death. That happens in the disease which is caused by Corynebacterium diphtheria and causes diphtheria. It causes a pseudomembrane formation on the throat, and the child usually, which is small, and if it's unvaccinated, they can die because of respiratory failure, because this pseudomembrane causes epiglottitis, and it can be very fatal. Okay, now important bacteria exotoxin. This is a picture similar to it is found in your Levinson, where we have different bacteria, and they're producing toxins. Here we can see Corynebacterium diphtheria produces diphtheria by means of the toxin which inactivates the elongation factor by ADP ribosylation. So there the mode of action is also given, and if the toxoid vaccine is possible to make or not. This is a very good graph. Try to go through it and remember it, because it will cover a lot of questions which are usually asked in the exam. Botulinum toxin causes botulinum, and this mode of action is by blocking the release of acetylcholine by proteolytic cleavage of releasing proteins. Then is Clostridium difficile, which produces pseudomembrane colitis. Usually it's produced when a lot of broad spectrum antibiotics are taken up by the patients. It leads to C. diff infection, and in this case exotoxin A and B inactivate the GGPases by glucosylation. Clostridium perfringens is another Clostridium which produces by means of releasing toxin which produces gas gangrene, and alpha toxin again is the enterotoxin, and it also acts as a super antigen. Then Bacillus anthracis causes anthrax or inhalational anthrax, and in this one, edema factor is an adenylate cyclase, also a lethal factor, which is a protease. It cleaves the MAP kinase, which is required for cell division. So these are the mode of actions. I know it will be overwhelming to see this kind of tables, but try to remember these because this mode of action will be different for each toxin. And the major thing is that they all are exotoxins. They are released outside the cell. Then in terms of gram-positive cocci, we have the already known bacteria Staphylococcus aureus and Staphylococcus pyogenes. Staph aureus secretes a superantigen which causes toxic shock syndrome by means of T cell receptor and induces also interleukin 1 and 2. So this is also present as food poisoning, which is caused by Staphylococcus aureus, in which it acts as a super antigen by acting on the GIT. Then the toxin of Staph aureus also causes scalded skin syndrome, which causes a lot of skin infections. And this toxin is a protease that cleaves the desmoglobin in the desmosomes. Then coming to Streptococcus pyogenes, it can cause scarlet fever, which is a super antigen again. Among gram-negative, we can see E. coli, Shigella, Vibrio, and Bordetella pertussis. These are responsible for secreting exotoxins, which cause major diseases like they can cause watery diarrhea, bloody diarrhea, dysentery in cases of Shigella. It can cause cholera in cases of Vibrio, and it can cause whooping cough or kali khansi in cases of Bordetella pertussis. So all these toxins have got different mode of action. So what happens in scarlet fever, which is caused by Streptococcus pyogenes? It happens. It's caused by the exotoxin, which is produced by lysogenic convergence, and this cytotoxin damages the blood capillaries and results in a skin rash. And also, it can cause strep throat with a rash. This is a picture of rash that the skin becomes reddish. So this is a disease caused by streptococcal toxins. This is scalded skin syndrome. It looks like burn marks on this hand of this baby, and also toxic shock syndrome, which is because of this super antigen. Now, the diseases which are caused by the neurotoxins are botulism, which is caused by Clostridium botulinum, which is a gram-positive anaerobic non-spore forming rod which is found in the soil. And also it works as a neuromuscular junction and leading to impulse blockage from nerve cell to muscle cell. And it results in muscle paralysis. These neurotoxins in the botulism, they are also used by plastic surgeons a lot for killing the wrinkles of the skin. You must have heard it कि botulism करवा लिया किसी actresses ने, and it can go bad as well. जो skin के ऊपर wrinkles आ जाते हैं because of aging, वो इससे treat हो जाते हैं. So the anti-aging effect can be utilized by inserting these toxins as well. But in actual, it can cause a disease which is caused by Clostridium botulinum, and it leads to muscle paralysis. The other well-known disease is tetanus, also called lockjaw. It's caused by Clostridium tetani, and it's a gram-positive anaerobic rod. It acts as a neurotoxin on the nerves and resulting in the inhibition of muscle relaxation. It causes tetanus. That's why when we have any accident, we always go for a toxoid so that this toxin-mediated disease cannot set in the host. If your vaccination is complete, then you can have a toxoid again if the time span after the last vaccination has been more than 5 years. So this is a classic picture of full-blown tetanus, which causes opisthotonus. Muscle spasms of tetanus are caused by neurotoxin of the Clostridium tetani. Now, what are the diseases caused by the enterotoxins? Enterotoxins are released by Vibrio cholerae, which leads to cholera, which is also called rice potter stool. It prevents conversion of ATP into the cyclic AMP. It causes cells to excrete chloride ions and inhibit the absorption of sodium ions, hence leading to watery diarrhea. There would be electrolyte imbalance because of sodium and chloride loss, and water leaves by osmosis. So there are two polypeptides in this toxin, in this cholera toxin, which is A active and B binding. Two parts of the toxins, and A is acting as an enterotoxin, which causes epithelial cells to discharge large amount of fluids and electrolytes. And in severe cases of cholera, even 12 to 20 liters of body fluid is lost in a day. That's why it's a serious disease, especially in children where it can be fatal. And mortality rate is about 50% if it's not treated in time. Usually one has to be given oral rehydration salt, ORS, and or IV rehydration if it's very serious. These are the rice water stool of cholera. And the A subunit of the enterotoxin causes epithelial cells of the of the GIT to discharge large amount of fluids and electrolytes. Now coming to another toxigenic disease is enterohemorrhagic E. coli, which is also called E. coli 0157H7. It's a very well-known strain of E. coli because it releases an enterotoxin that causes a hemolytic inflammation of the intestines. And it results in bloody diarrhea, or it causes dysentery. And this is a toxin-mediated disease, which is acting by means of altering the 60S ribosomal subunit. It inhibits the protein synthesis, resulting in cell death. Again, in this one as well, the lining of the intestine is shed, so it leads to, and along with some blood cells, so it leads to bloody diarrhea. So this is another good graph showing all the toxins and their effects. The endotoxin, which is a gram-negative lipopolysaccharide, which causes fever and inflammatory cell stimulation. While in different exotoxins, we have neurotoxins, enterotoxins, and tissue-invasive toxins, and pyrogenic toxins, and virotoxins, and they are secreted by different bacteria, and they have different effects. Like enterotoxin we talked about, like Vibrio cholerae, E. coli, Bacillus cereus, they all lead to diarrhea. While food poisoning, which is caused by Staphylococcus aureus or Bacillus cereus, can lead to vomiting and diarrhea. In tissue-invading toxins, we discussed Staphylococcus aureus, Streptococcus pyogenes, and Clostridium perfringens. They lead to tissue destruction by enzymes. And in pyrogenic toxin, which is the fever-causing one, it can again cause toxic shock syndrome by Staphylococcus aureus, or it can lead to scarlet fever if it's caused by Streptococcus pyogenes. Certain strains of E. coli secrete a toxin known as O157H7, which leads to hemolytic uremic syndrome, and it's called a virotoxin. And we also discussed Bordetella pertussis toxin, which causes whooping cough, Corynebacterium diphtheriae, which causes diphtheria, and C. diff toxin, which causes pseudomembranous colitis, which is usually by broad-spectrum antibiotic usage or in hospitalized patients. Again, another book picture from Levinson, which shows different mode of actions of the different exotoxins. Now coming in depth of the endotoxin. We are discussed already the exotoxin. Now we are going to talk about endotoxin. Endotoxins are part of outer membrane surrounding the gram-negative bacteria. And these are having a lipid A portion, which is the toxic part of these lipopolysaccharides. And the effects exerted when the gram-negative cells die and these cell walls undergo lysis, liberating the endotoxin in the host cells, and all produce the same signs and symptoms, which is chills, fevers, weakness, generalized aches, blood clotting, tissue death, shock, and even death. And this endotoxin can also cause miscarriages. The fever which is produced by these endotoxins, this is a pyrogenic response, is caused by endotoxin release. Again, another picture showing major differences between exotoxin and endotoxin. Endotoxin is part of the outer membrane of the gram-negative cells, and it is chemically a lipopolysaccharide. And it cannot be converted in a toxoid form, so ability to form a toxoid is not present in endotoxin. It's a heat stable molecule, and the mechanism of action of endotoxin is by innate immune response. A systemic response leads to fever, dramatic drop in blood pressure, and blood clotting leading to disseminated intravascular coagulation. And the toxicity is not as toxic for endotoxin as much as the exotoxin, but small amount can also lead to appropriate response that helps clear an infection. So this is the structure of a endotoxin, which is a lipopolysaccharide, having a lipopolysaccharide outer membrane and lipoprotein portion. So these endotoxins do not promote the formation of effective antibodies, and organisms which usually produce endotoxin are Salmonella typhi, Proteus species, Pseudomonas species, and Neisseria species, and all these are gram-negative, obviously. And medical equipment that has been sterilized may contain endotoxins. That's the infection control measures, that they have to be sterilized many times. And what events lead to fever by means of endotoxin? That these gram-negative bacteria are digested by the phagocytes, by our neutrophils. And this lipopolysaccharide is released by digestion in the vacuoles, causing macrophages to release interleukin 1. And when interleukin 1 is released, it is carried via blood to the hypothalamus, which controls body temperature. And this interleukin 1 induces hypothalamus to release prostaglandins, which resets the body's thermostat to higher temperature. Hence, the fever sets in a patient. Okay, this we discussed in the previous class as well, that the number of invading microorganisms are very important once they gain entry into the host cells by means of either mucous membrane or respiratory tract or genitourinary tract or conjunctiva or skin or parenteral root, whatever is the portal of entry. First they have to penetrate or evade the host defense by means of their virulence factors, whether they are capsules or any cell wall component, or by means of releasing enzymes. Then once they penetrate the host defense, they are able to damage the host cells, and they also insert cytopathic effect. They can be causing direct damage because of their endotoxins, or they can release toxins, whether they are exotoxins or endotoxins. These bacteria can also lead to hypersensitivity reactions. So, a little bit about normal flora of humans. I will not go into depth of these because they will be discussed later on, but just a brief statement that normal flora of the humans cause types of symbiosis called mutualism, which is a symbiotic relationship in which both the species benefit. The bacteria who is living on the human as well as the human cells, they both get benefit out of this relationship. And there can be commensalism, which is different, as this is a symbiotic relationship in which one species benefits and the other species neither help nor harm. So it is just harmless relationship for one host, and the other person gets the benefit. These are the types of symbiosis. And there can be parasitism as well, in which the relationship is in which one species is getting the benefit but the other species is getting harmed. That's why it's called parasitism. And generally the species that benefits the parasite is much smaller than the species that is harmed, that is the host. So these are usually the rules of normal flora. Normal flora is usually found on skin, upper respiratory tract, oral cavity, our intestine, especially our large intestine, and vaginal tract. These are already colonized with certain bacteria which live normally. So they are called the normal flora. And there is very little normal flora in our eyes and stomach because of the protective mechanisms of lysozymes and by the hydrochloric acid. And usually internal organs are devoid of any normal flora, and they are not there in the lower respiratory tract below the carina, like in the alveoli. There are no bacteria there. And in the muscle tissue and obviously our sterile fluids like blood, CSF, peritoneum, these are devoid of any normal flora. So no normal flora are present in there. Especially in pericardium and meninges, obviously, they are sterile. They don't have any bacteria. If some bacteria invade them, they can cause infection. So what are the benefits of normal flora? They provide nutrient production. They can lead to processing, for example, vitamin K production, which is by E. coli. It happens in our gut and normal intestines. And there leads to competition with the pathogenic microorganisms because these normal flora occupy the receptors and they protect us by not giving space to the pathogenic microorganisms. If the receptors are already occupied with these normal flora, the pathogens, they don't find any space to adhere and they cannot cause disease. And for normal development of the immune system, the presence of normal flora is very important. And normal flora can also lead to opportunistic infections if our defenses are down. If our immune system is hampered or broken down by means of any disease, whether it's any cancer or any infection like HIV, then it can lead to opportunistic infections as well. Now we'll come to generalized stages of infection. You must have heard since childhood that it happens, how disease occurs, but now we are going to formally learn the stages of infection. First of all, there is entry of the pathogen. Then, by means of the portal of entry, whether it's respiratory tract, GIT, skin, parenchyma, whatever is the portal of entry, first the bacteria gains entry in the host cells. Then it starts to colonize. It starts to multiply to live on that host surface. It is called colonization. And it is usually at the site of entry. Then there is an incubation period in which the bacteria keeps on multiplying and the patient does not have any symptoms or any clinical signs at this period. So this is an asymptomatic period. After the incubation period, which is between the initial contact when the microbe enters the body till the appearance of the first symptom. This is the incubation period. After incubation period, there are prodromal symptoms. What are prodromal symptoms? These are the initial symptoms. Then leads to final invasive period in which the full set of symptoms and disease sets in with increased severity. This is called the invasive period. And fever sets in, inflammation and swelling occurs in the invasive period of infection. And tissue damage also occurs, and this infection spreads to different sites as well. And after these 5 steps, there is decline of infection, which is the end or resolution of infection, which is leading to convalescence. Convalescence is usually the regain of health. So, another picture showing different stages in cases of acute, chronic and latent infection. First, there is incubation period, then there is illness, then there is convalescence. Convalescence is a time of recuperation or recovery from illness. Illness periods it will vary according to different microorganisms, and in this time, the individual may still be infectious and it can lead to spread of infections during incubation or convalescence. Okay, this is the picture showing different modes of infections like respiratory or salivary or respiratory infection spread by saliva. They can be spread of disease by fecal-oral spread in cases of GI infections. Some genetic urinary infections or sexually transmitted diseases are spread by the venereal spread. Then there are certain diseases which are vector-borne and in which the infections are acquired from animals, whether they are arthropods or it can be by insects as well. Okay, now we come to the second part of our lecture, in which we are going to discuss about the bacterial infection which are associated with cancer. Mainly, in the book we have discussed two main infections which are associated with cancers. One is the Helicobacter pylori, the cause of ulcers, the gastric ulcers, the duodenal ulcer. It can lead to gastric carcinoma and gastric maltoma, which is the gastric mucosal associated lymphoid tissue, MALT. MALT lymphoma. This is a cancer associated with this bacterial infection. Then the second one is Campylobacter jejuni, which is again a bacteria which can lead to MALT lymphoma of the small intestine, also known as the alpha chain disease. So these two bacteria are cancer causing and they have the potential to cause cancers. And this idea was supported because if these bacterial infections are treated earlier with antibiotics, then these cancers try to regress if they are treated early. So these are the main bacteria which are associated with causing cancers. Now coming towards the fact that what are Koch's postulate. These are the criteria which were produced around 100 years back that to ascertain whether the proposed organism is responsible for causing disease or not. So this guy Koch proposed certain postulates to establish whether any bacteria or any microorganism is cause of that disease or not. So the proposition was this organism must be isolated from every patient who is with the disease, and if the organism, it must be isolated free from all other organisms and grown in pure culture to establish that this is the one responsible causing the disease. And this pure organism which is produced in culture is able to cause the disease in healthy susceptible animal or a human, and this organism must be recovered from the inoculated animal. These four factors and are part of the Koch's postulate, and this leads to four discrete stages which we already discussed, that there will be incubation period, there will be prodromal period, specific illness period, the recovery period, and after recovery there can be cases of latent infections in only a limited infections. Latent infection is a hidden infection. It is a property of certain microorganisms which develop subclinical infection during which they remain asymptomatic, and the presence of the antibodies reveals that a prior infection has occurred in that organism. So, coming towards the end of the lecture now, what we did today among determinants of bacterial pathogenesis, we discussed the toxin production, types of toxins, exotoxins, endotoxins, their differences, and how they are causing disease by different mechanisms. And we also discussed the bacterial infections which are associated with cancer, like Campylobacter and Helicobacter. The different strains of the same bacteria, they can produce different disease, and we also discussed the typical stages of an infectious disease. So, hopefully these will be clear to you. Try to read up your book many times, especially the tables and the graphs which are given there, because many information can be taken from that, and especially making MCQs and scenario, we try to incorporate that. Thank you very much. Allah Hafiz.